Okay, now for the last section of Chapter 18, gene regulation in cancer. So, cancers, these uncontrolled cell divisions that result or can result in the formation of tumors, what we know as cancers, um, can arise for many reasons. Now, what happens is essentially there's a loss of control of the process of the cell cycle and cell division. That is, the cell is dividing uncontrollably. It's not paying attention to or listening to instructions anymore, you might say. Um, so these there are many genes, again, that we control the developmental process that control the cell cycle. And these genes that play an important role in the cell cycle that have the possibility of mutating and causing cancers are known as proto-oncogenes. And when they have mutated and are now in the state of promoting uncontrolled cell division or not preventing uncontrolled cell division, they're known as oncogenes. So proto-oncogenes become oncogenes, and once they're oncogenes, they are ones that um, will essentially promote cancer, if you will. Um, so um, these proto-oncogenes become an oncogene essentially through mutation, and how the oncogene does what it does can vary. Um, so these proto-oncogenes, again, that code for proteins that control the cell cycle. If for some reason they are a copy of them or the gene itself is moved to a new location in the genome and is being expressed at times when you don't want to express it, um, that can promote uncontrolled cell division. If the gene within the same chromosome becomes amplified, it can um, overstimulate the cell cycle. So now you're producing too much gene product, too much of the protein. So you can produce it at times when you're not meaning to produce it. You can produce too much. Um, so that's not necessarily a mutation, the gene itself, but it being in an inappropriate place through translocation or being multiplied, or amplified, I should say. And then a change to the gene itself, a mutation in the base sequence, um, either within the gene itself or in one of those upstream control elements, can cause the gene product to be made again at an inappropriate time or too much, or the form of the gene product can change such that now it either becomes hyperactive or it just is not doing its job properly. Um, so again, these proto-oncogenes control the cell cycle, and again, you can either have some that promote the cell cycle that become modified, or those that inhibit the cell cycle and become modified. So the an example we have of one that promotes the cell cycle is what's called a RAS gene. And so it's essentially responsible for making a protein that stimulates the cell cycle. All right, so here we have, and this is some of this stuff we're talking about here is things from chapter 11 that we kind of need to revisit. We haven't done that yet, um, but we'll talk about that some. So we have these proteins embedded in the membrane, and their recipients, they receive this growth factor. And they initiate a series of steps that stimulates one of these RAS proteins, and the RAS gene is responsible for making this protein. And the RAS gene sort of propagates the signal into the nucleus such that a gene is expressed that stimulates the cell cycle. Okay, if this RAS protein, if the RAS gene is defective and therefore the RAS protein is defective, it has the potential to initiate this process in the absence of the growth factor. Normally, you have to have the growth factor present, but the defective protein can just do its job anyway in spite of the absence of the growth factor. So then you're going to get this cell dividing and that forming this growth, this tumor, when that's not what you want to happen. All right, an example of a tumor suppressor gene is P53, and P53 produces a protein that normally binds up the DNA and prevents expression of genes that are involved with the cell cycle. Um, However, a defective form of this P53, and they're showing here, it can perhaps be mutated by something like exposure to UV light, which can damage DNA in your skin cells and lead to skin cancer. So if this tumor suppressor gene is mutated and its gene product is defective and it doesn't bind up the DNA when it's supposed to, again, the cell cycle can proceed at an inappropriate time. So it's not suppressing the cell cycle. So again, these are sort of the two ways these oncogenes can function. They either can overstimulate the cell cycle or not inhibit it when they are supposed to. Okay? Either case, you can result in inappropriate and increased cell division in a tumor. Normally, cancers take time to develop and multiple genes to be modified, mutated to end up in cancer. A good example of this is colorectal cancer. This is something that tends to strike later in life, and so it's thought that, of course, as one gets older, you have lived longer, you've been exposed to more things in the environment, and in general, there's just simply been more time for mutations to occur, which increases the chance of cancer. Colorectal cancer is in your rectum and colon, and this tube is part of your digestive tract, and what can happen is these little growths known as polyps can develop on the inside of your colon, for example, your large intestine, and they can get bigger and still be relatively benign, but then they can start to break through the wall of the colon and spread and become malignant. At about 50 years of age is when people start getting colonoscopies, where a gastroenterologist, a GI physician, will take a look up there and look for these things and remove these benign polyps to prevent them from becoming malignant. And again, about age 50 is when you start doing this, because that's really the time when these start showing up. Now, so exposure to things can cause mutation, but you can also just simply inherit mutated genes. And there's a particular gene called the APC gene. If you inherit a mutated form of that, it can promote colorectal cancers, and about 15% of colorectal cancers are due to this, so you inherit it. The others are from exposure to things through life. And a relatively small proportion of breast cancers are due to these particular genes, BRCA, BRCA1 and BRCA2. These are both tumor suppressor genes that, again, in the mutated form, don't suppress the cell cycle. And I put a picture of Angelina Jolie up here because she was in the news several years ago because her mother had breast cancer and maybe even died of it, I don't know. And she had herself tested, and it turns out she had a form of one or both of these genes that essentially is the oncogene form. So it's a cancer promoting form, and I remember hearing that they were saying she had about a 85% chance of having breast cancer. And so because of that, she had a mastectomy to remove that tissue, which essentially eliminates that chance. And so it was a sort of a big deal at that time. Here's a link to a website at the National Institute of Health, I believe, that you can plug in any kind of gene and get any kind of information. So if you go there and put in APC or BRCA, you can get all sorts of information about these genes and their normal functioning, and then what happens when they are not functioning normally, when they're mutated. Okay, that's it for chapter 18.